Started here, the final session for the day on harmful algal blooms. Glad to see such a great turnout. I've been looking forward to this session. I'm Tammy Zimmerman. I'll be the chair for this session. I'm from the USGS. And our first speaker today is Dr. Campbell from Mercyhurst University.
It's nice to be here, and I'm glad there are so many people interested in harmful algae. It's something that I became interested in when I was doing my master's work in the Poconos and did some plankton work and saw some cyanobacteria blooms in some little recreational lakes in the Poconos. And that may have been at some level what triggered me to want to take a sort of a regional look at the cyanobacteria blooms in our area here this summer. I've got several things that I would point to as what led me to do this project. Dr. Diz and I have been working with our students at Gannon and Mercyhurst respectively for the last 2 years with a grant through Pennsylvania Sea Grant and NOAA to develop a model of the cyanobacteria bloom phenomena in Presque Bay, and Dr. Diz will be speaking more about that. But Angelia, who is one of my co-authors pictured here, on the Environaut, Gannon's boat that we've been sampling from, this summer kind of took a big step and was doing pretty much all of the sample analysis. After having learned how to do phytoplankton analysis last summer, she did the zooplankton this summer too. So that gave me a little bit more time to play since she was able to handle the processing of the samples. And it was intriguing as the summer unfolded. There seemed to be a lot of press all around the country about harmful algal blooms, not just in the Great Lakes, but in places as far away as California and Maine and Minnesota. And the news report in mid-July indicated that we were expecting the bloom in lake algae to be pretty big, maybe not as big as it had been back in 2014, but a big one. The other thing that inspired me to do this particular thing were the weekly emails I was getting from Amber Stillwell reporting on our microcystin toxin levels and other toxins that she was measuring here. And even though I never replied to any of those emails, I did look at some of them. And this particular one caught my eye because I didn't realize that they were sampling places besides the waters of Lake Erie and Presque Isle. When I saw Eaton, I thought, hey, you know, that's an interesting lake and it's not too far away, and you know, I've got a couple of hours this afternoon. Maybe I'll go out and grab a plankton sample. And it helped that my son, who's a John too, has a little bit of history with plankton sampling, and he's the one that actually suggested that maybe we go out to some of the lakes and get some plankton samples that we could compare in late July, what we were seeing in all the lakes in the region compared to Presque Bay. So on July 27th, we started with Eaton Reservoir and then Findlay Lake, since it was right down the road. And I lied to John. I told him that Lake Pleasant was on the way home. And as the crow flies, it wouldn't have been too far, but it's not actually too easy to get from Findlay Lake to Lake Pleasant, as I found out. But we sampled Lake Pleasant, and then over the next week, we found time to visit as many of the lakes that we could. We wanted to kind of hit most of the natural glacier lakes in the area, as well as some of the larger reservoirs. And my son is teaching me about technology. He created a website that we could use for posting our results. And if any of you are interested, we've got a website called harmfulalgae.com where you can go to get all of our results. And you'll just see sort of the tip of the iceberg today with this presentation. So we sampled all the lakes once initially within the 8-day period. And Lake Pleasant and Canadota we decided to sample again because the densities of cyanobacteria we found in late July were so low, we just wondered if maybe it was an unrepresentative sample. So we sampled those two lakes again. We sampled from publicly accessible locations. If there was a dock or a boat launch area that allowed us to get out into deeper water from shore, we took most of our samples from piers and docks. And we used a 65 micron mesh Wisconsin style plankton net and a 5-meter long horizontal tow. I would have liked to have done oblique tows or vertical tows, but since typically the water wasn't much deeper than a meter or two, it made more sense to go with horizontal tows. For Chautauqua Lake, which we went to last, we decided since it was so big that we took two samples, two different sites, one at Long Point where there had been a report of high microcystin levels in late July, and then we went down to Jamestown and took a sample at another park northwest of Jamestown. Presque Bay, we didn't do extra sampling there since we had already been going out on the bay every 2 weeks with Dr. Diz and the Gannon students to do our regular sampling as part of the NOAA Pennsylvania Sea Grant funded study. But the data that I used here were the data for 2016 and 2017 in the same window of time in late July as the samples that we collected from the other lakes. And we have got 3 sampling stations there. I'm sure Rick will give you a little bit more information about the particulars of this study, and Angelia will also be sharing some of her findings with the phytoplankton and zooplankton. Our laboratory processing was pretty standard. We'd look at the samples live and take pictures, and my son got into doing the microphotography of the algae, and most of these pictures are his. And after looking at the samples live, we'd preserve them in 5% buffered formalin, and then took a 1 milliliter subsample to count at 100 times magnification in a Sedgwick rafter counting chamber, and then calculated the population densities in original lake water in units of numbers per liter. The data provided some interesting snapshots of the different types of variations in plankton numbers among and within lakes. The Lake Erie Presque Bay data here kind of shows July 27, 2016 results compared to this summer's results. And interestingly, the total densities were lower in late July this year than they were last year. But Microcystis, which is the taxon that has been often associated with the production of the microcystin toxin, had almost double the densities this year. But in general, we had a much more vigorous bloom of other cyanobacteria species last year at the end of July than we saw this year. The samples from Chautauqua Lake were particularly instructive because it gave us an idea of how much sample population densities of cyanobacteria might vary between 2 different sites on the same lake on the same day, with several orders of magnitude difference in the densities at Long Point compared to the site near Jamestown. And most of those big differences were due to one genus, Aphanizomenon. The Chautauqua Lake site was interesting too because these large spherical colonies of trichomes and gliotrichia were visible to the naked eye. And I had no idea because I had not seen very many of these before in a plankton sample. So we were kind of excited to see what those balls were when we saw them in the lake. To give you an idea of the regional variation in 2 of the taxa that seem to be most widespread among the lakes, this graph shows in orange bars the microcystis densities, and in blue bars, the Anabaena species, of which there were several different ones. And notice it's a log scale, so it doesn't really give you as accurate a picture of how much things varied among sites. But I believe these two taxa are ones that are commonly associated with some of the types of toxins that Amber ended up measuring, and I'm sure we'll learn more about that from her. So the Anabaena was definitely the genus of cyanobacteria that seemed to be most widespread in our regional lakes, and there were a handful of lakes that had very low or non-detectable levels of microcystis. When you take the microcystis data and take out the log scale, you can get a better appreciation of the actual differences in densities among the sites to compare them. And I singled out microcystis because of its relationship with the microcystin toxin production. And sure enough, Presque Bay topped the chart for all the sites that I measured during this narrow window. And I'll be curious to see, with the difference between last year and this year in microcystis, if Amber has any data on how her microcystin levels compared this year to last year, too, in the bay. Aphanizomenon is a good example of why you need to look at samples live. The photograph on the top is Aphanizomenon in a live sample where the trichomes are often in a bundle of sticks which zigzag back and forth against one another. And then when you preserve a sample in formalin, it usually breaks up those bundles of trichomes, and then you find the individual trichomes as in the diagram at the bottom. And this particular cyanobacteria is the one that seems to be most capable of generating really high population densities in the lakes. And interestingly, Findlay Lake, which was one of the lakes that seemed to have the largest densities of cyanobacteria, has still got a large bloom of Aphanizomenon as of last week. So unlike the bay, where the cyanobacteria blooms seem to dissipate by September, we've still got a booming bloom out on Findlay Lake. When we resampled Canadota Lake, it was interesting to see how much the cyanobacteria population densities changed in just 2 weeks, with the Anabaena taxa in particular blooming during that period. And a similar thing, only even more dramatic in Lake Pleasant between late July and 2 weeks later in August, with an Aphanizomenon bloom that really took off, but also the Anabaena again. Microcystis, fairly low densities in Lake Pleasant and not as much of a dramatic increase. So what I conclude here is nothing all that remarkable. There's no surprises here in what I'm saying, but the cyanobacteria bloom formation in our lakes in this part of the country is apparently a seasonal, regional phenomenon that is understandable on the basis of how these things respond to temperature changes on a seasonal basis and light availability. But it was kind of interesting to me that the same taxa were showing up in all of the lakes at about the same time, even the lakes like Lake Pleasant and Canadota that have better management and control of their nutrient inputs. There are large variations in the cyanobacteria population densities within large systems from year to year, as in Presque Bay, and from one site to another on the same date, which we could see from the Chautauqua Lake data. And we've got so much data now on site-to-site variation in Presque Bay, we could provide lots more examples about how variable within the bay the plankton numbers are. And finally, because of what I saw in Canadota Lake and in Lake Pleasant, I think it's important to mention that any kind of ranking of regional lakes based upon single date sampling is probably not very reliable. especially now that I see Findlay Lake still blooming in late October and early November. It's possible that some of these other small lakes might have pretty dramatic blooms later in the season than what we see in Lake Erie and in Presque Bay. So I want to give special thanks to Rick Diz, who now that he is retired is now an apple cider engineer, and he's teaching me his craft. And we're going to have a taste testing episode soon where we see who made the best tasting hard cider with his apples. And Sean Rafferty and Pennsylvania Sea Grant, I thank Sean Rafferty and God that our funding through NOAA actually came through again this year, despite doubts about federal budget. And Rick, that's your grandson.
Yes, I recognize.
What's his name?
Graham.
Graham. I always enjoy our trips out on the Environaut, and I wish in some ways that I worked at Gannon University for that reason, but I'm okay with Mercyhurst. I've been there 34 years now, so I guess I'll hang out with them yet. So if you have any questions, I'd be happy to answer them. Yes.
I can tell you at Chautauqua, the Pennsylvania is still going 2 weeks ago.
Is it?
Yeah.
Wow.
This is the North Basin, but on the other side, on the eastern bank of the, or sorry, the western bank, more on the north end.
Yeah.
It was the chunkiest I've ever seen any of Pennsylvania blooms ever. It's intense. But it's really great to see that diversity across all the different lakes. Did you compare any of the counts to the water quality parameters or environmental?
I did not. This was purely biological. You know, it's nice to kind of work without the constraint of a grant. You can do whatever you want. Whatever you don't want to do, you don't have to do. Yeah.
Could you give us an idea of how these numbers and these lakes compare to Western Basin, Lake Erie? I mean, like how much? Is it threefold?
You know, I would imagine that some of the higher densities that I've reported here are probably comparable to what they're seeing in the Western Basin. And I have not made direct comparisons, but some of the aerial photography, satellite imagery of the bay, you know, compared to the Western Basin, it's got a similar look. So, I mean, the bay in a way is like a little mini version of the Western Basin because it's shallow and it's more directly affected by the nutrient inputs that are coming into it. So, but I honestly don't know. I haven't looked at the population densities in the Western Basin to compare. That would be an interesting thing to do, yeah.
So there's no water quality or nutrient data with this thing?
No, there is not. I have a lot of information about the lakes, though, and there's some nice straightforward explanations as to why Canadota Lake is so clean and Findlay Lake is so not clean. And it has to do with all those cottages that are connected to a sewer system at Canadota Lake and not connected to a sewer system at Findlay Lake. So septic tank leachate, I'm sure, is one of the big factors contributing to the strong bloom every year at Findlay Lake. Until they can do something about the septic systems, they'll probably see it or it'll get worse.
We have time for maybe one more question.
I do have one from the stream, actually. Can you speak more to the temporal variability you saw? Was it on the scale of hours or days?
The temporal variability in Canadota Lake and Lake Pleasant was 2 weeks difference, which I tried to look at weather data to see if there might have been something to account for that. If we had really warm weather in those 2 weeks, and it wasn't. It was actually unseasonably cool. There were a couple rain events, so it could have been, you know, a little input of nutrients that caused the surge. But even, I mean, one of the storms was a ½ inch of rain, and the other one was a 20th of an inch. You know, so it wasn't enough rain to really account for a big surge of nutrients.
Thank you, Dr. Campbell. All right, our next speaker is Dr. Rick Diz from Gannon University, and he's going to be presenting on nutrient loadings from the watershed.
And as it happens, I've got a different version of this file.
Oh, I'm sorry.
No, it's not your fault. I just loaded it earlier when I came in. There we go. I feel very constrained by the microphone and is it battery? Yeah. Oh, excellent, excellent. Because I just hate standing behind that thing. So, so you heard my name mentioned several times already, and one of the real joys of being a research scientist in Erie, Pennsylvania, is getting to work with Mike Campbell. I think we started working on stream projects 15, 18 years ago, and I look forward to every session where we got together to go out on that boat, and every other time that I've had the opportunity to work with Mike. I don't think I've ever known anyone who took such joy in collecting samples. And I just find it so invigorating and inspiring. So, but I only have limited time. Who holds up the...
That would be me.
Oh, okay.
I'll give you a 2-minute warning.
I appreciate that. Okay, thanks. As Mike explained, he and I are partnering on a project that's being funded by the Sea Grant program, and of course we're very grateful for that opportunity. It's a long project, and there are several component parts to it. Dr. Campbell is the biologist and is responsible for and in charge of analyzing those samples that we collect out there for the organisms, and now I understand that he's foisted that off on Angelia, that it's really her job. Maybe I'll get some of those numbers sooner than I did last year as a result of that, Mike. You already have some numbers. That's good. I'm the water quality guy, the hydrology and water quality guy, so we complement one another's areas of interest and expertise quite nicely. And I'm limiting my talk really today to nutrient inputs from the watershed. And I have only a little bit of information about what we did in the Bay. We're still a long way from having any real conclusions. This is one of those images that, that Mike referred to. This is probably the most recent image that NOAA has posted on their website about HABs. It's really interesting, isn't it, to see this bloom of, we assume microcystis, that they sort of claim that that's microcystis. I find it quite interesting that the flow coming down the Detroit River here is sort of pushing it away, and it's almost like a flame coming out of a Bunsen burner kind of thing. Just the sheer force of the water apparently is pushing that algal bloom away from Detroit, which of course is right there, and is apparently fed by the Maumee River, which comes into the lake right there and deposits large amounts of nutrients that mix with the very nice high-quality water coming down from the upper Great Lakes, and then in this sort of soup pot kind of environment leads to these blooms. This is what is this little bay right here? Sandusky Bay, yes. And in passing, they always mention, well, Sandusky Bay has a bloom. They don't seem to be very interested. Unfortunately, these satellite images don't tell us much about our, but this is another graph that's presented on that NOAA. that now has a bar column for this summer season, indicating the relative severity of this year's events combined, consolidated, compared to prior years. We see this disturbing trend in the western end of Lake Erie, where it appears that things are getting worse. And of course, none of us are happy about that. And we certainly don't want blooms of these organisms in Presque Bay either. So we are similarly concerned. The question about temporal variation and Dr. Campbell's observation about how much difference it makes from one day to the next. This is from, as you can see, from the summer of 16. This is chlorophyll concentration derived from the DEP's buoy that is moored in Presque Bay from about early May through the fall, through September sometime. And as you can see, the remarkable and dramatic changes in chlorophyll concentrations from sometimes dramatic and extreme changes just from one day to the next. It's really quite remarkable. I never expected to see that kind of behavior. And so we had, this red is the water temperature of Presque Bay. That buoy also records water temperature, several other parameters it also measures. And it's been very valuable to me in trying to provide a backdrop for what we're doing. So, so here we have a bloom of something that's green, another little bloom, bloom here, it goes back down, goes back up, and then look at that. That's just incredible. That's essentially all on just a single day. Numerous measurements. Of course, this records a data point every 6 or 10 minutes or 20 minutes or something like that. 20 minutes. Yeah. Now, this doesn't tell us what's causing that, but it's something green. That's all we really know so far. So the goal of our project is to try to understand the factors that would lead to a bloom of harmful algae. That sticks in my throat a little bit. Cyanobacteria are not truly algae, but we tend to call them that. They're photosynthetic bacteria, very primitive organisms. We have several parts of our project. I'm going to talk mostly about my study of water and what it contains coming into Presque Bay from the watershed. We care about nutrients a whole lot in trying to understand this aquatic ecosystem because we assume that there are only certain factors that are limiting. And nutrients, and when we use that term, we mean nitrogen and phosphorus primarily because they are scarce and often one or the other limiting to the growth of photosynthetic organisms that get their energy from the sun. And the response of cyanobacteria compared to the true algae is complicated and not entirely understood just yet. And it might be a number of ways of looking at nutrients that control the appearance of these sudden blooms of organisms and then their sudden disappearance. It might be an interaction of several factors, and that's why we're hoping to use an ecosystem, an aquatic ecosystem computer model sponsored by the US EPA that hopefully will help us organize and synthesize the information we've got. Temperature is certainly important. Folks like to dispute the issue about climate change. I don't know what's going to happen 10 years or 100 years from now relative to the water temperature of Presque Bay, but that might be a very important factor since cyanobacteria have an optimum growth rate at warmer temperatures. And the other bacterial group, algal groups, typically, generally, prefer cooler water temperatures. This is that same chlorophyll graph, but in this case I've overlaid a few of the data points that Dr. Campbell provided to me. For certain of our trips that we made out into Presque Bay last summer. This is in milligrams per liter, biomass in milligrams per liter, not counts. Cell counts and biomass concentration are different because some of those cells are very small, some are big, so just the number alone doesn't tell you everything that we might need to know in terms of nutrient uptake and growth rates and things like that. The purple dots here are cyanobacteria. These brown squares are diatoms. Traditional thinking says that diatoms don't like warm water temperatures, but here we are at the end of July, early August, and we've got what appears, apparently this bloom right here, perhaps a combination of cyanobacteria and diatoms. We would expect that this out here would be diatoms because diatoms seem to do very well in cool temperatures. One other interesting thing here is that the so-called green algae, which I thought would have been more abundant, very low levels, at least in this data set that I presented there. So I'm going to describe what we did in the few minutes I've got left to monitor the streams coming into Presque Bay from its watershed. This table shows the more important streams and their length and the area that they contribute as a percentage of the total watershed. I had monitoring equipment on these two streams, Cascade Creek and Mill Creek, which together constitute about 75% of the watershed. This is a little map that gives you, for those of you who aren't familiar, gives you an indication of the Mill Creek watershed. That's, remember, about 50% of the Presque Bay watershed. And here we have the Cascade Creek watershed. Most of this is actually in storm sewers. Very little of this is in open streams, except in the very lower limits here, and then in spotty little bits and pieces here and there. This is the sort of monitoring equipment that we used. This is a box located alongside Cascade Creek. As you can see, it was taken last winter. This is inside the box, a big 12-volt battery, and this ISCO sampler. This is all gone now. The storm on Sunday with the tornado and the huge rainfall, almost 3 inches of rain in about an hour or so, completely washed this box away. It has disappeared. The box is gone. The boulders, you remember the boulders? We sort of lodged it, gone. All those boulders are gone. I could not find the equipment. I went downstream searching for thousands of dollars worth of scientific equipment, and totally gone. So in order to quantify the loading of chemicals or substances into the bay, we have to know how much water there is, and then we have to know what's in the water. So the monitoring equipment simply measures depth. You then have to convert depth to flow rate or discharge, which is volume per unit of time. And that all by itself was a huge challenge because this data set is complicated. But these are the stage discharge relationships that I developed for each of the two streams. These are based on something called Manning's equation. If any of you know any hydraulics or hydrology, these are based on Manning's equation, then sort of factors that are consolidated and simplified so that I could use them a little bit better. One of the complications is that the monitoring site for Mill Creek, because it's in a tube, for those of you who know the area, Mill Creek is in this huge pipe until it emerges at the Bayfront Highway down at the wastewater treatment plant. So if we wanted to get the flow through the city, we had to be below the mouth of the tube, which meant we had to be down at the wastewater treatment plant, which is very close to Presque Bay. Well, Presque Bay goes up and down all the time. Presque Bay and Lake Erie go up and down all the time. It's sort of like a tide, but it's driven by winds. It's called Seiche behavior. And this up and down, up and down, up and down. So this is an overlay of Presque Bay or Lake Erie's level fluctuations along with the same data set from my monitoring station for that same time period. So figuring out what the flow rate in the stream is when the water's going up due to Lake Erie going up, and then down when Lake Erie goes down, was quite a challenge. So I had to figure out a way to subtract out the influence of Lake Erie on the monitoring station. This is a storm event. You can see these peaks where the blue goes up quite a bit more than the green, sort of anomalously it goes up. That's a storm event. That's a storm event. But here we had dry weather, so the monitoring station just followed the lake level, essentially. Rainfall is the driver of all this, and it's quite variable. And so this blue line shows the average normal rainfall in Erie during, if there ever is a normal summer. The sort of rust-colored bars are the summer of 2016, month by month. You can see it started out dry and then it got wet at the end of the summer. And then this past summer, we had a very wet June, including another huge storm event at the end of, well, the middle of June. Then it got quite dry. So the hydrology of the streams is driven by the rain, which was quite different for the 2 summers that we have been studying. This is a summary. This is just for the summer season, June to September. These are the results of my calculations for how much water left each of these streams. I made an assumption, 2 minutes. I made an assumption that the Cascade Creek watershed was representative of the unmonitored part of the Presque watershed, and just prorated that quantity to represent the unmonitored portion. So when we had almost 19 inches of rain during that summer, my calculations show that we had 13.7 million cubic meters of water leave the watershed and go into the bay. But this past summer, with less than 15 inches, it was about 11.5. The water per square mile is interesting. We're getting more water per square mile out of the Cascade Creek watershed than we are out of the Mill Creek watershed. That sort of makes sense because the Cascade watershed is much more urbanized, and the Mill Creek watershed has a higher percentage of what we call pervious surface, where water can infiltrate into the soil. I pointed those numbers out there. This is just a history of storm events and the discharge in Mill Creek and Cascade. That red line is the rainfall. You can see the rainfall and the flow in the stream correspond quite tightly with one another. This is for this past summer, where we had these very sharp increases in flow in the stream, and then it goes right back down again. These are very urbanized streams that have very flashy behavior. Now, we also measured nutrients and other substances in that water. The ISCO samplers were programmed to activate when the water level rose. So for the number of storms that we were able to collect samples and analyze them, in 2016, we only had 4 storm events that we were able. This is in addition to measuring water from the bay, where we went out every 2 weeks and we're analyzing bay water. We were also trying to analyze stream water. So we had 4 events for Mill Creek and 4 for Cascade. For some reason, they weren't exactly the same ones. Phosphorus is the thing that we mostly focus on. This is TP is total phosphorus. Total phosphorus is more than just phosphate. It's all of the forms of phosphorus in that water sample. The dissolved total phosphorus number is after that water sample was filtered. So the particles are removed. And so you can see 0.34 milligrams per liter in Mill Creek and 0.3 in Cascade Creek water. Nitrogen may also be important. We don't know that yet. I tried to focus more on the streams this past summer, and so we were able to get more storm events. And now remember, the rainfall was different. So there was less rain, so maybe the chemicals were, were more concentrated, 0.37, but this is a strange number. This jumped all the way up in Cascade Creek to 0.56. I double-checked that several times. This is just a summary to more readily compare those. I can't quite stop. I have to keep going. So to better understand where the phosphorus is, this shows the blue is the dissolved phosphorus and the brown is the particulate phosphorus. So you can see that in both streams, about 3/4 of the phosphorus is attached to particles. This is an individual. This was all the storm events. This is an individual storm event with samples collected every 10 minutes during that storm event. You can see that the dissolved concentration stayed pretty much the same throughout the entire event. But the particulate concentration went up and came back down again as the water became very, very turbid, and then as the storm event was ending, became less turbid. We then used concentration and flow together to calculate mass. And so these indicate the mass loadings of these chemicals to the bay. And notice they're very much storm dependent. So if someone can figure out how to trap the particulate matter during storm events, Amber was going to figure that out, that would be great. Because it's mostly during storm events. That's a huge undertaking. This is another way of looking at those numbers, and I show here the production rate per square mile of phosphorus. Notice that Mill Creek produces a little bit more per square mile in 2016, but less per mile in 2017. Remember, the amount of rain was different. This also has to do with that very high concentration during 2017. Nitrogen, less rain, higher production per square mile. Very strange. I don't understand why. This is a schematic diagram of the aquatic ecosystem computer model that we're going to use to try to assimilate all of this information. And we still have lots to do. Dr. Campbell and Angelia are working on the plankton. I'm going to be working on the aquatox model, and then once we get it to work, whatever that means, then hopefully we'll be able to speculate by doing so-called what-if scenarios. Sorry that I couldn't stop.
While we're loading up the next talk.
Yes.
Are you taking information about sort of wind forcing that isn't storm related when it comes to looking at changing cyanobacteria populations, right? So you saw that flow coming out of Detroit pushing the cyanobacteria out. Is there to any degree that wind forcing can cause sort of daily changes?
I think without a doubt that the wind causes local accumulations of the surface scum. These organisms grow in the water, but they produce gas vesicles, and they are buoyant. It's one of their adaptations. So they float to the surface, they get lots of light as a result, and then they get blown around. So there's an easily visible result of that. You see the scum, and it gets blown to one shore or another shore, it gets trapped in a little cove or what have you, and you can see that. And that's bad as far as human or dog exposure, but they're growing in the water. The model that we use will treat Presque Bay as if it's a complete mix reservoir. Wind is an input to the model, but I don't think it's not even a two-dimensional. It's not a three-dimensional model. It's just going to calculate one set of numbers for the whole.
So what do you think about that, like, one-day spike?
I have no idea. Ask.
All right.
I have no idea.
It could have been a wind event.
Water chemistry doesn't change that fast.
Right.
Biology can maybe change that fast, although I think that's amazing. Maybe something was wrong with the sensor. Where are our DE people? I don't have any explanation, but the water chemistry doesn't change that quick.
All right, thank you. All right, our next speaker is Angelia Belfiore. She's an undergraduate from the Mercyhurst University.
Hello, everyone. My name is Angelia Belfiore. I'm a junior environmental science major at Mercyhurst University, and today I will be talking about a concentrated aspect of this big project that we're doing with respect to rotifers and their population dynamics with respect to cyanobacteria blooms. So a little bit of background. We didn't really get into the details of this because we didn't, but for my project, or for this specific presentation, I'm talking about rotifers, so here's an introduction to some of the rotifers. In terms of what is in the bay with plankton, phytoplankton or zooplankton, we have the diatoms, green algae, dinoflagellates, etc., which I'll get into later because that will show more of what is important with the results. But with zooplankton, you have grazers, predatory, and so with the focus of this, you have Caratella colaris, Polyarthra, and Caratella quadrata. And so I'm focusing mostly on the middle one, the Polyarthra, but they all play an important role with respect to the total planktonic community. So the parameters of the study, Dr. Diz and Dr. Campbell both kind of talked about it. It's to kind of a predictor model, but I'm not really, that's not seen to fruition yet, so I won't get into it because I'm not really sure of all of the variables that go into it. And so to hone in onto the rotifer life, they are significant with the total planktonic community because of their biomass and their numbers. They are dominant because they are the link between the primary producers, the algae, and the bacteria, which aren't primary producers, and the zooplanktivorous fish, as well as the predatory zooplankton, such as Leptodora, the copepods, Daphnia, etc. So for our sampling location, we also talked about this previously, but Presque Bay, and as it was mentioned, Presque Bay, which is not as similar to Lake Erie, it's much shallower, and therefore has a lot more variability in terms of the temperature from a day-to-day basis. So on this is just us out on Dr. Diz's sailboat when we couldn't take the Environaut one day, but we're collecting just temperature and light readings. And then this is from one of us on the boat. And then these are the 3 sampling locations for which we took our samples. And so some of the methods, and so from last year, from May to October, every 2 weeks we would go out, and then from this year, every 2 weeks from June to September, taking triplicate samples at each of the locations. The first year we took vertical and horizontal tows with the 5-meter plankton, 65 micron mesh, Wisconsin style plankton net. And this year we decided to do vertical because we saw the results and realized we could hone in on just the vertical to get a better reading of the communities. At each location we would take set sheet depths, light and temperature readings along the water column. And then, of course, we retrieved the samples. Compared to our sampling last year to this year, we decided to not use a preservative this year than we did last year. So in this picture, you can kind of see it. We would go out in the field with our 10% buffered formalin in each of the jars, collect the samples, and then put them in there. This year, we decided that we didn't really need to do that, and I could process the samples within a week while they were still alive, and that would be that. So in the lab, these are some of our samples from this year, and I'll get into the differences in colors and such. So each sample was enumerated via a Sedgwick rafter chamber, and it was a 1 milliliter sample of each of the 9 samples that were taken per week. They were reduced once all the samples were counted and accounted for. They were reduced using a plankton splitter, you know, one of those things where you turn splits into 2. And those were 200 milliliters, and then 1 sample was preserved with a 95% ethanol alcohol, and this one was 10% buffered formalin. And you can see the difference between the 2 samples, not only between the May and the August, how much more is just in here compared to into here, but also with the color and the pigment. And that's why we wanted to do, or we wanted to process the samples live to see, you know, just how biology works, how it all moves together. And I think that was probably for the best. And so, with what we saw this year as well as last year were diatoms. So Fragillaria, Asterianella, Melisira, which was the #1 diatom that we saw. For green algae, Pediastrum simplex as well as duplex. And then, of course, the cyanobacteria, Microcystis aeruginosa, Lingbia, Linetica, as well as other species, Anabina spiroides, but also Anabina wisconsin and Anabina floss aquae, and Aphanizomenon. And some of the zooplankton species, we had both cyclopoid copepods and calanoid copepods, Asplankna, Leptodora, as well as the rotifers, but these are more predatory, so I figured I would include them in. And now we get to the results. Very interesting, very unpredictable because of the variability between the past year and this year. The numbers are kind of all over the place, like just this one goes up with the cyanobacteria, but that was Caratella erlineae, and this year the numbers are very small. Now there's a lot of speculation as to why that could be, as well as why Polyarthra this year are all over the place, and last year they weren't, or they weren't as much right here. So I'll get into, I guess, the reasons and the reasoning for why that is, but just to take in the entire variability, not only of the zooplankton, but also the cyanobacteria. And when I'm talking about the cyanobacteria colonies, it's just all of the totaled of those previous 4 or 5 species that I just discussed. And so I thought it would also be interesting to include the predatory zooplankton that I had mentioned in the other slide. So we have the cyanobacteria again, cyclopoid copepods, Leptodora, and the Asplankna. And I thought it was important to, of course, include the error bars, but you can see how every increase and decrease, they are almost always together, which is a very interesting, but very, just kind of out of the blue. You're not really sure why, yet, why that was happening. But to include the Polyarthra species spiking at 200 right when everything else was decreasing makes you wonder why. So there are a few speculations that came up with. So there could be a relationship between the microcystin levels with the rotifers. So as other large zooplankton such as Daphnia and calanoid copepods have a negative correlation with them, Bosma and other rotifers have a positive correlation. The presence of certain rotifers, as I mentioned in the 2 comparing graphs, their presence last summer compared to this summer could be the result of the presence or absence of the differing levels of the toxins. We're not really sure yet how to monitor that, but we could continue to do lab work in a controlled environment where you're the one, the scientist is the one who's introducing the toxins, the levels, and what species is exposed to what level. But because of the 2-year study, we were able to see from those the numbers were going to be normal every year, or if it was just by chance that there was a spike in a certain population species. And so for further works, these are all very zoomed in pictures of Microcystis aeruginosa. And you can see the little bacteria or little filaments around it stuck in the mucilage. And that raises the question if the rotifers, such as polyarthra, are feeding on the bacteria, and those are the bacteria surrounding the microcystis, then perhaps their populations are spiking because of the increased amount of bacteria with the clusters of Microcystis aeruginosa. And it also could affect the amount of bacteria could be affected by the stage in which the colonies are either living or starting to decompose. So as they start to decompose, more bacteria are going to be surrounding them, and therefore there'll be more available nutrients for the rotifers, polyarthra, to be feeding on. So that is just one way in which we can continue this study and continue to look at things. This is just the tip of the iceberg when it comes to the information that we've gathered over the summer. This is just a very specific species that we've looked at, and there's a lot of different outlets we can go to. We took temperature, light readings, and those could play into all of this as well. But because of the time constraints, not only of this speech, but of getting all the information, because our last sampling date was September 21st, there's a lot of information to just kind of digest and see where it all leads to. But there's definitely a lot more work that can be done. So a thank you to the Sea Grant for funding from Michigan University, Dr. Campbell, Dr. Diss, Trevor Sturgeon, who was the man pictured in the first picture with the beard, and to the TREC and the Regional Science Consortium. So are there any questions? Yes.
Are you doing any measures of something like heterotrophic plate counts or something like that that might help you get at the question of whether that kind of primary production of bacteria is related to the spikes you're seeing in some of the grazers?
We haven't done any of that. This is just kind of looking at a more like larger planktonic communities, so like the algae and the zooplankton, but nothing lower than like the bacteria, because that's just, I guess, not within the scope of the project. But definitely something to consider. Yes.
Angelia, with the, since you have live samples that you can look at, you can see those rotifers in there spinning away, doing their little thing.
Right, yes.
Can you actually observe them feeding?
Well, what's interesting is with the rotifers, they are always surrounding themselves around, a bundle of something. And they're moving through and kind of like grazing over it, which is what they do, they're grazers. But with others, there's a lot going on within the sample, so it's hard to focus on just one small individual organism when there's a giant copepod swimming by and moving everything, But it's definitely something that you notice, because those are the ones that, are swimming, but the rotifers are the ones that are honed in on a certain clump of something and are focusing around that area.
But you're pretty sure that they're not feeding on the microcystis?
Oh, no, they're not feeding on like the actual microcystis cells, but perhaps what's around them in like the mucilage.
It's way too much of a mouthful for them.
Yes.
I know you mentioned this, but I'm just thinking back. How did you sample? Was it vertical or horizontal?
Vertical. Yeah. The first year we did vertical and horizontal, and then we did some testing, and we realized, like with the numbers that we got, we realized there was no significant difference between the two. So we realized with a vertical tow, we would be getting a better, I guess, representation of the whole water column, and like instead of just the horizontal aspect of the first meter of the water.
I think there's one more question in the back. Does somebody have their hand up there? All right. Thank you very much, Angela. All right, our next presenter is Amber Stillwell from the Regional Science Consortium, and she's going to be telling us about the research she's been doing with flow cytometry.
Hi, everyone. For those that don't know, I'm the laboratory manager at the Regional Science Consortium, and I had the pleasure of working with Mike and Rick for both my undergrad and my graduate work, so that was very, very fun. So today I'm going to talk to you guys about something that I've been working on all summer. It was also funded by Sea Grant, so thank you, Sea Grant, for funding this project. I use something called a flow cytometer to measure cell counts of cyanobacteria. So first I'm going to take you through what a flow cytometer is, because I'm pretty sure maybe no one in the audience knows what it is or is unfamiliar with the way it works. So this is the flow cytometer that I use. There are multiple different kinds, but this is the BD Accuray C6 Plus flow cytometer. And this is it as it sits on a desktop. And this is it on the inside. And I have zoomed in pictures of the inside later. So what the flow cytometer does is it uses red and blue lasers to collect a fluorescence signal from cells of algae. And this can be used for multiple different things. You can use flow cytometry to collect data on blood cells, red and white blood cells. And also I've read studies where they have collected samples and counted diatoms and all sorts of other things. So what I was particularly interested in was looking at the natural fluorophores that cyanobacteria give off as they go through these lasers. And those fluorophores include chlorophyll, which we've all heard of, but then two others, phycocyanin and phycoerythrin. So these cyanobacteria have these natural fluorophores, or a natural fluorescence. Phycocyanins are what are measured from our buoy. So like Dr. Diz showed the chlorophyll readings on the buoy, we also have a probe on the buoy that measures phycocyanin. So I also was able to measure phycocyanin in the cells using the flow cytometer. And this is all important because it goes through how I was able to actually obtain my data. So phycocyanins fluoresce at around 650 nanometers. So this list down here shows you all of the different light filters. And up here, there are 4 light filters on the inside of the flow cytometer. And those light filters essentially are picking up the fluorescent signals that are bouncing off of the cells as they go through the red and blue lasers. So to pick up phycocyanin, you have to use light filter FL4, and that is able to pick up wavelengths of 675 nanometers, plus or minus 12.5. And you can see FL4 here, and that is the filter, and then the fluorochrome that is actually inside the filter. And so we use the fluorochrome APC, and this will all be related later, I promise. It's a little to take in, or a lot to take in. The second important fluorophore is phycoerythrin. So they fluoresce at 488 nanometers, and so we had to use the FL2 light filter detector on the machine, and that is fluorochrome PE. So after reading a ton of studies and doing a ton of background research, I realized that the best way to look at microcystis as it passes through the lasers is by comparing the phycocyanin to the phycoerythrin, therefore giving me these axes of the light filters, PE versus APC. So these are a couple of the studies that I read showing the results that they found. So there are multiple variations of each fluorophore. And you can see up here all the different fluorophores that were analyzed in that one study. And then down here, this middle picture, you can see that the flow cytometer, the same model that we use, was actually used to collect data on different kinds of algae in the water, including diatoms and chlorophytes and cryophytes. So this piece of machinery can be used for many, many, many different things. It just depends on where those things fluoresce and if they have a natural fluorescence and if we're able to detect that. So down here is a study that I read that was able to look at Microcystis, Synococcus, and Anabaena species all separately using the flow cytometer. And those axes that I mentioned before are here and here. So we have our phycoerythrin versus our phycocyanin. And you can see that all of these cyanobacteria are fluorescing in around the same area, but they all have varying mass sizes. So these are density graphs, and you'll see my particular density graphs later, but this is just showing you where that colony of, or where that species of cyanobacteria is fluorescing on your graph from your machine. So the machine is feeding all of that information to the computer model, and the computer model gives you this as it is being processed. So the density shows you the highest density of points of the cell. So that's where most of the cells are accumulating. But then you have these other cells around the outsides that may be dying off or may have a slightly different fluorescence than their, the average cell. So this is a depiction, well, it's actually a screenshot of a standard of Microcystis that I ran through the machine. So after I figured out how to configure the axes, I was able to run a couple standards. Well, actually, I ran a lot of standards before deciding that this was the way to go. I also was able to get input from BD Biosciences, who created the machine, and they were able to tell me exactly how to gate around the colonies that I was looking for. And what that means is, so this here is everything. That is all of the data that was collected in my standard of Microcystis. And I had to adjust those axes to find what I was particularly looking for based on its fluorescence. So that's what this image is here. And you can see it's about, it's almost 100% Microcystis in my sample. And that makes sense because it's a standard. So from there, after you gate around this, you can actually label this and change the color so that it will show up in any of your other graphs. So this is a histogram showing one peak, and that one peak correlates to this, or corresponds to this coloring, this gating that I did around my standard of Microcystis. And like I said before, this is a density graph. These all three at the top are density graphs, and then these are population graphs. So they're just the number of events that occurred, and you can see the masses there in red and purple, and I'll get to what that red means later. So after I was able to figure out how to read Microcystis in the using the flow cytometer and to figure out how many cells per milliliter of Microcystis were in each sample, that was my next step. So what I had to do was take all of my data, each sample I ran in duplicate, and they all went through the same process. You can adjust the settings here. You can change the size and the width of the needle that the cells are pulled up through the lasers by. You can also adjust the flow rate of the machine to change how fast it's pulling things through there. So after I figured all of that out, I ran every sample in duplicate. And after that, I would run a standard. And those standards, each one I would gate around, and then you can project that standard's location as it fluoresces onto all of your other samples and get percentages of the cell count of the cells that you're looking for. So from my original standard that I showed you guys, it was almost 100% microcystis. I was able to run or to compare those cell counts or those gating areas to samples from, these are particularly from the marina on Presque, and they're from the same sample, so Marina A and Marina B, so they're my duplicates. And we have about 7% microcystis in the A sample and 6.9% microcystis in the B sample. And I also wanted to show you guys both sets of samples just to show you how close together the machine is counting those cells. All of my percent CVs were very, very low, so I consider that very good. So the other part of this is knowing your debris. So environmental samples are dirty, and barring a way to filter samples but keep my microcystis cells in them, I had to figure out a way to get the debris out of my samples before I can filter the samples. Because believe it or not, it's very difficult to filter microcystis out, or everything but microcystis out of a water sample, because microcystis is relatively large. So what you essentially have to do is look here in the very, very front corner of your sample. So this is my standard again, and this is that teeny tiny dot that's circled in red is debris. And I know that because when I can add these up together in the computer program, it tells me that this is, let's look exactly. I think it says 97.9% microcystis. So this would make up the rest of that chunk of the debris and what's not in my standard. So it could be dead cells, it could be different chemicals in the water that are being read by the machine, but we have to know what that debris is and where to look for it so that we can remove it from our analysis when we're looking at our dirty environmental samples. And yeah, I can also gate around that, and that I have in red. So if we look, this is my standard sample, and if you look at the histogram, that shows just the microcystis because the debris is actually so small, it's not showing up on our histogram. And for my Marina A sample, that is also projected onto here, and you can see all of those populations in red, all of that debris, and it does show up on our histogram here. But just a little bit of that purple there is our microcystis. But luckily, the machine allows us to batch analyze all of our samples so that we can actually only look at what we want to look at. So after I gate around that microcystis, this is the debris gating and this is the microcystis gating. I can choose to only analyze the microcystis graphs from all of my samples all at the same time. So if I select this graph and select all of my samples, then I get a long list of all of my statistics. But I can actually export all of that data to Excel or to PowerPoint, which is very, very convenient. And that was where I was able to take that data, pull it off the computer, and then do my post-processing to figure out how many cells per milliliter of microcystis that I have. So getting into my results, the whole goal of this is to try to figure out how cell counts of microcystis correlate to to the microcystin toxin. Turns out that is extremely difficult, as you all probably know. So we analyzed the toxin by using the Abraxas ELISA kits, and we also did the quick lyse beforehand on all our samples. We did them in duplicate. After the results were read by our microplate reader, we put them into the macro embedded data analysis worksheet, which is it's given to us by Abraxas themselves and helps us calculate the exact micrograms per liter of microcystins that we have in our samples. So then we can take those cell counts from the flow cytometer and compare them to the toxin concentrations that we got from the same sample on the same day. So these are our sampling locations. We actually had 24 sites around Presque Bay, along the Presque beaches, and on Lake Erie. This year we did something new. We actually extended our sampling locations all the way from Elk Creek towards the west, almost to the Ohio border. and all the way out to Freeport Beach, which is near the New York border. So all of these sites they enabled us to really broaden our database and see what's going on in more areas of Lake Erie, as well as all of our sites inside the bay and along Presque Isle beaches. So 6 sites along the beaches and then 12 sites in the bay. We also additionally processed water samples from Erie Waterworks and Northeast Drinking Water Authority. And Erie Waterworks brought us their drinking water samples, and Northeast Drinking Water Authority brought us their samples from Eaton Reservoir, as Dr. Campbell mentioned before, and also several other of their drinking water reservoirs, as well as their raw water intake from the lake. I should mention that we didn't actually do flow cytometry on the drinking water samples. We're hoping to have the funds to do that next year so that we can do cell counts versus the concentration of toxin. But for the most part, the samples from Erie Waterworks have very, very low concentrations of the cells and the toxins, so we didn't see the need to do that. I wish, looking at Dr. Campbell's data this morning, or this afternoon, I wish that we had done Eaton Reservoir because we would have had some really interesting cell counts to compare. So I said already that we sampled our locations, but we did it once a week, every Thursday. And we took the samples basically the same way that we take a bacteria sample. So you wade into the water waist deep, and then you stick your arms in elbow deep, unscrew your sterilized bottle, and then screw the cap back on and take it back on to the lab and make sure that it's chilled in the process. All of our samples were processed within 24 to 48 hours, depending on what staff we had and how much time we had at the end of the day. I mentioned that we did the quick lyse. We did each quick lyse sample in duplicate, and then we did our cell counts for cyanobacteria in duplicate as well. And we had 20 total sampling days. We actually did 21, but we only did 20 sampling days where we processed both toxin concentration and flow cytometry for the cells. And this is one of our interns, Julia Guerin, doing an ELISA test over the summer. She was learning, and she did a very good job. And this is one of our signs indicating that there's toxin levels above dog recreation threshold. So at that beach, or actually that's at Shades Beach in Northeast. So that day I took that picture. I was like, this is really good. I can use it for a presentation someday. So this was taken this summer. You can see it's very, very green. And I actually took this sample from the beach, believe it or not. Beach 6, we had a really, really bad bloom this year. And unfortunately, I don't have the data yet to compare. I have all the data, but I haven't sat down and compared the two from 2016 to 2017. But this was the first summer that we had a massive bloom on the beach side that was visible, lapping green waters up onto the lakeshore. So that day we had to go out and help the lifeguards determine whether or not they should pull people out of the water. Okay, so these are preliminary results, and the reason I say that is because a lot more work has yet to be done on this data set. So I'm just going to show you guys the flow cytometry results versus the microcystins results. So flow cytometry is indicated by the letters FC, and then MC indicates microcystins, the toxin concentration. So up here you can see the R data. So I did correlation charts for all of our samples, and I can answer questions about those afterwards. But these two in particular I wanted to show you. This was one of our highest correlations. So it's a pretty strong correlation. You can see the peaks moving up and down with each other at the marina on Presque Isle. But then at another location called Boaters Beach, you can see this peak here of the cells is completely opposite of the toxin concentration. So that's what's giving us our really low correlation. These are for the Presque Isle Bay samples. You can see all of our correlation values there. I was a little disappointed with some of these because places like Vista 3 always have really high toxin levels, but it didn't have as high of cell counts and not at the same time as indicated up here. And I think the reason for that may be because we actually collect that sample almost right in the morning, so at 7 a.m. or maybe 20 minutes after. And as Dr. Diz said earlier, they do have those gas vesicles where they can propel themselves up for the sunlight. So I'm thinking maybe that we have to change our methodology there so that we're getting sunlight exposure when we take that sample. And these are from the beach. We had some pretty strong correlations, especially at beach, well, beach 11 was the only strong correlation. And then we have a very weak correlation at beach 2, which is interesting because that beach exceeded its values almost all summer for dogs. And then our East Erie samples, we had some high correlations at Freeport Beach, and at Mill Creek, we took this sample at the wastewater treatment plant location. However, we only have about 6 weeks of data for this, so it's a smaller data set. So I'd have to build that data set a little bit more next summer in order to consider this valid. And these are our West Erie samples, and as you can see, we did not have very strong correlations in West Erie, but at Elk Creek we did. We actually had an exceedance at Elk Creek one time for dogs, which that value is 0.2 micrograms per liter, by the way, so it's very, very low. That site was just very interesting because it's right where the water is very slow moving, and I think that it collects a lot of interesting things in it. Just to wrap up really quick, I know that I only have about a minute, but I think that one of the problems with our collection, and maybe why we don't have such strong correlations as I expected, is that we only sampled once a week. So as you guys probably know, the cells can be there maybe on Wednesday, but then they're gone by Thursday when we take the sample. So we're scooping that water, we're taking the sample and analyzing it, and we're getting those high toxin concentrations, but not necessarily high cell counts. And then I also need to look and see the comparison between the other toxins that we analyze, which are anatoxin, saxitoxin, and cylindrospermopsin, because microcystis can release multiple different toxins, but my other issue is that the flow cytometer right now, as I'm running it, cannot differentiate between cyanobacteria. So I did a couple trial runs before the summer started. I combined Anabaena species with microcystis, and it did not look any different. I was not able to find a density that was significantly different than the other. So that's something that I'm going to work on this year, and trying to run different types of cyanobacteria through there and see if I can get a better handle on splitting those genera apart, and hopefully that will enable us to have a better ability times with our cell counts if I can get us there. So again, thank you to Pennsylvania Sea Grant, Great Lakes Restoration Initiative, and the PADEP, specifically the Coastal Resource Management Program, for funding this research, and also to BD Biosciences for helping me analyze some of my data, and to Jeanette Schnars for supporting this project, and also to all of our interns who worked really, really hard this summer to collect all the samples. And any questions? I know.
So one of the, it's great, you saw some really good correlations. Do you think that the fact that you're lysine cells has any effect on the relations that you're seeing? So if you just looked at extracellular toxin, do you think that some of those that you didn't see correlations at might tighten up?
Yeah, that's a really good point. We should definitely look at doing that for next year because, yeah, you're right. If we are lysing all the cells, we're getting our maximum toxin concentration. So, yeah, that might not necessarily be, yeah, thank you.
I can't remember, was there a pattern, was your cell count always lower than your toxins when you had those, when you didn't have correlation?
Mm-hmm.
Was there one always higher than the other, or was it a mix?
It was kind of a mix, yeah. Some of the correlation graphs show really, like the patterns are moving together. Generally, I think the cell the cell count was higher than the toxin concentration, just by looking at those, because obviously they're on different graphs. Yeah, but at some points, we did see the toxin concentration exceeding the cell count. But then other times when, we had zero correlation, like even a negative correlation, it was, high cell counts 1 week, and then at the end of the summer, high toxin concentration, and just, yeah. So I need to look more at the general area. I want to look at the general characteristics of those areas to see maybe if they're shallower or if they have warmer temperatures generally, because we collected a ton of other data that I just have yet to dig into.
I'm just wondering too, if it's sort of like almost kind of opposite, like if it's a legacy, kind of where the cells have moved through, but the chemistry is remaining or something.
Right, yeah. And we do have some nutrient. Yeah, we do have some nutrient data that we collected from the stream locations that we process. So hopefully we can get that analyzed, and I want to make that comparison as well and look at that to see. Did you?
Well, tagging on to those ideas, if you're on the leading edge of a bloom, then perhaps, total speculation, you've got a lot of young cells, relatively speaking, that have not lysed or exported toxins to the water. If you're on the trailing edge of a bloom, you might have a lot of old cells that are coming apart and releasing the toxin to the water. So you could have the same number of cells on the leading edge versus the trailing edge and have a very different physiological state for those cells. Does that make sense?
Yeah, it does. Yeah, and I want to, if we can, someday sample more than 1 day a week so that I can try to compare, if maybe on Wednesday,
You're already.
I know, and I may be even able to look at last week compared to this week.
With Dr. Campbell?
Yes, I did.
I can tell.
Okay, thank you.
All right, next up we have my colleague Joe Duras from the USGS, and he will be presenting on some of the cyanohab studies that we have going on within the USGS and the Great Lakes.
Thanks, Sam. So, Dr. Diz, I'm gonna follow you. I can't stand behind the lectern. I can't do that. Thanks, Tammy, for the introduction. Thanks for having me here to talk today. So I'm gonna kind of take this because Amber just had such a great talk, and then we all asked her questions about, well, what about this piece of data and what about that piece of data? I'm going to kind of give an overview of the activities of the Great Lakes Restoration Initiative, the USGS Harmful Algal Bloom team. And we're usually pretty particular about this. We usually call them cyanohabs when we talk about them. So they're cyanobacterial harmful algal blooms. That's specifically what we're interested in. The way that I'm going to go through the talk today, I'll kind of give a little bit of an outline, and then I'll talk a little bit about the processes and kind of how we think about things conceptually. And then I'm going to talk about the data that we collect, and I'll end with a few examples of some of the data analysis that we've done. So to move in first, I would be remiss if I didn't mention our team. This is not just me by a long shot. In fact, I'm kind of on the lower side of this. I manage a lot of the data, and I help to make sure that people have what they need. But these are the group of folks that really make this go. Marianne Evans is in our Great Lakes Science Center, which is in Ann Arbor, Michigan. Carrie Givens, Lisa Fogarty are in Michigan, and Pete Lineker is in Wisconsin, which is now part of our USGS Upper Midwest Water Science Center. So we're constantly getting shuffled and renamed. Erin Stelzer is in our Ohio, Indiana, Kentucky office. She sits in Columbus, Ohio at the Ohio Water Microbiology Lab. James Larson is an ecologist from USGS Upper Midwest Environmental Sciences Center. And Keith Lofton is he's an engineer by trade and a chemist by passion, and he works at our USGS Kansas Water Science Center at the Organic Geochemistry Research Lab. So as you can kind of tell as I went through, all those people have kind of very different views about these issues. Some people are really concerned with how the toxins are cycling and how they're forming. Other people are really concerned with bloom initiation, what's driving bloom formation, what's driving bloom senescence. It's one of the things that we don't think a lot about, but we're really interested in what makes it go, but we also want to know what makes it stop. And then some people, Carrie and Aaron in particular, are very focused on the microbiology and the microbial interactions that are happening within these blooms. So this has been said, I think, in every talk, but I'll say the same stuff. You know, we have these cyanobacterial harmful algal blooms. They've been occurring annually in Lake Erie, and they've been getting worse in the last 15 years or so. The severity of those fluctuates year to year. Severity can mean two things. It can mean the kind of extent, so how big the bloom is, but then also the density within that extent, so how dense the cells are there. USGS has been conducting research around the basin as part of the GLRI for a number of years, since about 2014. We're just in the process now. We feel like we've got enough of the variability hammered down that we're starting to evaluate data and look at it across multiple seasons. So where have we been looking? Well, primarily in the western basin of Lake Erie. We're there with about, I think, 5 other federal agencies and about 20 universities evaluating what's happening in the western basin of Lake Erie. But in addition to that, we felt it was important, because we were being funded by this Great Lakes restoration and not western Lake Erie restoration, to really make this kind of a broader Great Lakes study. The core of our team kind of sits in that central area in and around Michigan and Ohio. So we also evaluated the Saginaw Bay. Oh, that's not the right button. Laser, there we go. We also evaluated the Saginaw Bay. We evaluated the Grand Traverse Bay. And for a methods comparison, we evaluated Green Bay in Wisconsin. If you know anything about these bays that I just described, obviously we have a very eutrophic bay, we have a mesotrophic bay in Saginaw Bay, and an oligotrophic bay in Grand Traverse Bay. Green Bay is very seasonally eutrophic during the summer, much like the western basin of Lake Erie. It is thick with microcystis. What we did in these locations, we've been just doing like very routine monitoring down in the western basin. We did a trophic state comparison study in 2015 where we evaluated it and used the exact same methods on these 3 bays. And then again, as I said, this was a molecular method comparison that we did up in Grand Traverse Bay, or in Green Bay, because one of the things that we're very concerned with is you have a lot of people doing this research and everybody's using different methods, and we want to say what NOAA says is similar to what USGS says, is similar to what NASA says when we do all this information. So, a conceptual model. I like this model because it shows just how complicated it is, and it's in fact only really begins to scratch the surface. So it's a very simple cartoon. So what this cartoon indicates here is, you can see you've got a succession of organisms in the water column, right? You've got algae, you might have some microcystis cells, you have some diatoms, and then with the addition and changes in temperature and carbon dioxide, in nutrients coming in and out of the system, and nutrients coming from different landscapes, you get the succession of bacteria and of algae and zooplankton and all of those things, and that comes into play. And you eventually get to a tipping point in the population where you get enough microcystis, that's your cyanobacteria du jour, that's primarily what we're focused on, that you get a bloom forming. Okay, so once the bloom forms, that's not the beginning or the end of it, that's kind of the middle of the process in a lot of ways. Those of you who look at some of that buoy data, you might see that the pH changes on a daily basis. That's very driven by the photosynthetic processes that are happening within the bloom. As the bloom photosynthesizes, right, it's taking carbon dioxide out of solution. The pH then begins to go up in the lake. That has an effect on what metals are in solution, so it begins to affect solution chemistry. There's a lot of important things that are happening there. There's a kind of a fight for carbon dioxide as the bloom gets more and more dense. Dr. Diz and Dr. Campbell both mentioned temperature as being a very important driver of these things. You have to get to a certain threshold temperature before you're really going to see cyanobacteria take over. And then at some point, and for some reason, and I'll make that statement loudly, toxins are produced. One of my favorite ecological questions is, why do they make the toxin? They don't make it to poison dogs or to poison people. They certainly do it for some ecological reason. There are a lot of hypotheses out there about why that is. One of those hypotheses is that this toxin actually might be a way that the cells are communicating with each other. Another hypothesis says that it potentially has something to do with metal sequestration, so that they can get rare trace elements that are hard to find and they need to scavenge in the bloom. Yet another says that it might protect them from over intense UV light, so it's some sort of large sunscreen molecule. All right, so there's a lot of ideas out there about what those molecules are. And then, of course, you have this bloom senescence, so maybe you get a viral infection, right? We don't think about that. The bloom gets sick. Maybe that's what causes the bloom to senesce. And then cells deposit. Some of those cells are alive, some of those are dead. And then you might get wave action that resuspends that back up, so you have this kind of ongoing cycling process. And then certainly there's other things that we need to think about. We need to think about nutrients coming out of the soil, right, or out of the sediment. When we get anaerobic conditions, we know that dissolved phosphate will oftentimes rise back into the system, and that can affect things. We also know that, that nitrogen can pulse out as ammonia from certain anaerobic sediments. We have the effect of secondary producers. So you guys might know this, there's 1 or 2 zebra mussels in the western basin of Lake Erie. We know that they filter the water, they clarify the water. That clarity leads to deeper photopenetration. It seems like, based on a few experiments that have been recently published, that zebra mussels and quagga mussels can preferentially feed on green algae, meaning they're choosing to eat green algae and spitting out cyanobacteria, enriching the cyanobacteria in the water column. And then external to that, we have these weather inputs, right? We've got the days where it's sunny, we've got days where it's cloudy, we have days where there's wind, where there's stream inputs that are really high. So all of these things affect what's going on out there in the lake, in these big lakes. And it's important to keep in mind that these are complicated systems. And the way that we have to address this is we have to collect really enough data to start to understand these. So with that, I'll talk a little bit about our sampling program. So we do consistent sampling. It's ranged for the tightest range that we've done is daily for about 3 or 4 day periods. And the longest that we've done is about a 2-week interval where we're sampling every 2 weeks. What we do when we sample, we take an integrated photic zone water sample. So we integrate the entire photic zone of the water column. And then we take another water sample where we capture water from 1 meter from the bottom. In addition to that, annually, at the beginning of each season, we sample sediment with a ponar. We also filter water, and we collect those for DNA analysis, for both qPCR analysis and for metagenomic sequencing. We use a plankton net to do plankton counts and biomass, as has been described previously. We take field parameters every time we go out. We look at pH, dissolved oxygen, conductance, temperature, turbidity, dissolved CO2. We look at chlorophyll A and phycocyanin, and then we do second-disc transparency. So no matter what, you always take your second-disc with you, no matter how much other stuff you're collecting. As far as our analytics, well, we do some pretty intense chemistry. We use the USGS National Water Quality Lab, and then also some boutique analysis that we like to call it boutique when it's somebody that does one analysis. Somebody does one analysis in our Wisconsin center, that's Pete Lineker. At the Water Quality Lab, we do full suites of dissolved and total nutrients. So we look at nitrogen, we look at nitrite, nitrate, ammonia, total nitrogen. We look at orthophosphate, and we look at total phosphorus. We also look at total metals and ion chemistry. Because we know that the blooms are affecting carbon dioxide in the water, that means that they're affecting the buffering capacity of the water. We know that, we see the pH changing, right? So that affects ion chemistry, and we think that there might be, there's some suggestions in the literature that some trace metal chemistry can actually play a role in how and when the organisms start to produce toxins. So we look at things like sulfate, chloride, alkalinity, iron, lithium, molybdenum, and zinc. All of those have been implicated as potentially being important for toxin production. In addition, we also look at dissolved organic carbon. So not unlike what Amber was just describing, the flow cytometry, you can do the same thing on a bulk sample, and what that tells you about is you filter your sample, and then on that bulk sample, what you do is you look at it and you excite it with a UV light, and then it gives off some other UV light in response. And what that tells you about is the quality of the dissolved organic material that is present. The reason that's important is because as these blooms are growing and moving, we know that they're producing and exchanging molecules. And one of the things that we're interested in is understanding how the quality of organic carbon might be influencing either when they die or when they produce toxins. We do toxin chemistry. This is done mostly at our USGS Organic Geochemistry Research Lab, although we've done some work with the Michigan Department of Community Health as well. We typically use ELISA assays, which is the process that Amber just described. We look for microcystin LR equivalents. We do both totaled and filtered microcystin, and we do that pretty much on every sample. Now, at the beginning of this, back in 2014, we were really interested, we're going to find all these other toxins out there, and so we sampled for saxitoxin and antitoxin and cylindrospermopsin, and we never found any of them. We looked in all across all those sampling sites in the Great Lakes. The only place we found any other toxins was in Green Bay, and that actually wasn't USGS doing the analysis. That was the folks in Wisconsin that we were working with that found that. In addition, on some samples, we've done LC-MS-MS. The reason this is important, you may or may not know this, the ELISA assays that we typically use are focused on this LR, but there are, I think, a growing list of 30 or more different congeners now of microcystin. Those congeners actually cause this test to respond differently. It's not a one-to-one response. So microcystin LR will give you a unit of 1. Other microcystin variants might, instead of giving you a unit of 1, give you a unit of 3, or a unit of 0.5, right? And because there's mixtures of these things in the blooms, we feel that it's important to get past just using the ELISAs and start to look at the individual congeners and to see how those come into play. To that end, Keith Lofton in our Kansas office is really interested in understanding the precursor molecules. Unlike a lot of proteins that bacteria make, these bacteria, these cyanobacteria, don't use this and package it in a ribosome. If you were here this morning, you might have heard about our DNA and our RNA. That's typically how the ribosome is how bacteria make proteins and fold proteins. Microcystins are assembled and folded extra-ribosomally, right? So they make these big molecules and then they fold outside of the ribosome, and that's kind of a unique process. So understanding these little precursor molecules might actually be important to understanding the system. Algal and related analysis. You've heard about a lot of this already, so I kind of dropped through this. We do microscopic cell counts with a contract lab. They do algal scans. They identify algae and cyanobacteria, and they enumerate cells per milliliter. The Great Lakes Science Center, where Mary Ann works, they do total biomass, ash-free dry mass, and chlorophyll analysis. We do have samples preserved to do phycocyanin extraction analysis, but we haven't found a very good method. So if anybody out there is doing phycocyanin analysis on extracted filters, I'd love to talk to you. And then microbiology, we're doing enumeration. For this, we use quantitative polymerase chain reaction. I'm not going to get into the details of that, but suffice it to say that we can target individual pieces of DNA from particular organisms and then count those genes in the environment. We do that both for DNA, which is kind of an indication that it's there, and then for RNA, which is an indication that it's actively producing that gene in the environment. So we measure total cyanobacteria, total Anabaena, Planktothrix, total microcystis, and then we measure the toxin forms that produce microcystin of Anabaena, Planktothrix, and microcystis. And then we measure a gene for the production of saxitoxin and for anatoxin. As an example, this is just a gene diagram of the different complexes of the MCY gene. So, and then lastly, and maybe the most complicated thing to understand, I guess I left the best or the worst for last. So the Michigan Bacteriological Research Lab for the USGS, they characterize the bacterial community structure in sediment and in water. They basically, if you think about it this way, that I just described quantitative PCR as targeting an individual gene. What this sequencing process does is it basically targets every gene that's in the sample and sequences it all at once, and then you get back on the order of 3 million different sequences. You kind of have to sort through those with some advanced computing, but what that lets you know is it lets you start to understand the proportion of how things like Anabaena might relate to Microcystis, might relate to Synechococcus, might relate to some of the other cyanos. And we're advancing that now to the point where we're including 18S into that, which will then tell us about how the things relate to green algae as well. All right, so we do all that data, right? And as you can imagine, we have this conversation a lot. We need more data. Yeah, we always need more data, right? We have that conversation a lot. But we're now to the point, like I said, where we're starting to analyze some of this. So I'll give you just a couple examples of the way that we're looking at this. One of our initial questions was whether the sediment might be seeding the blooms. So to orient you, Michigan, Lake Erie, Western Basin, Detroit River, there's that nice smile that Dr. Diz was pointing out. Maumee Bay, right there is the river raisin. That doesn't get talked about a lot, but it could also be an important source. So at the mouth of the Detroit River, what you'll see here is, these are all collected on the same day. You see that Microcystis is very low, Synechococcus is a relatively higher proportion, and then there's a bunch of total cyanobacteria that we're not even catching what they are. That has to do with the quality of the libraries we use. It turns out there are a lot of cyanobacteria out there that we don't have any way to compare to because they've never been sequenced before. So we have a lot of unknown cyanobacteria. Now compare that with Maumee, where you have Microcystis in very high quantities in the sediment, right? So this is in the spring, before there's any bloom activity. This is just a ponar grab of the sediment. And you see that the relative abundance of the cyanobacteria is greater. Compare that to the Toledo intake, which is right about there. And you see that it's different again. There's no Synechococcus here, but the abundance of cyanobacteria are very similar. And then you can see that there's diversity across the basin. So, very complicated plot. This is my last one. So I'm right there. The thing I want to point out here, so these are dates, and those are the same 3 cyanobacterial species. So this is blue is Microcystis, red is Synechococcus, which is a picoplankton, and then the green is total cyanobacteria. We have cyanobacteria qPCR data and Microcystis qPCR data. So this is general cyanobacteria. That's Microcystis specifically. And then we have the biovolume. So one of the big differences that I want to point out, this is from W1 and W2, so these are on either side of the river raisin. You'll notice that on let's see, where is it? It's the 721 sample to 8.4, we see this shift where Synechococcus drops off, right, and Microcystis shoots up. Here we see Microcystis come up, but Synechococcus doesn't go completely away. So even within this kind of closer spatial proximity, we're seeing differences in the cyanobacteria community, and those tend to be related to things where we're seeing like large increases in biovolume. So what that's saying is that Microcystis is suddenly taking off. We're getting big increases in biovolume. And we have plots and plots of data with nutrients and carbon and all of those other things overlaid. Eventually, we're going to start to do some ordination analysis to really start to pick apart what might be driving 1, bloom formation, and 2, toxin formation. So here's some publications. James wins the publication thing. He's got 1 in press and 1 already out. The rest of us are still scrambling around in preparation. So I'd be happy to answer any questions.
We're running about 7 minutes behind, so.
What does that mean?
Yeah, 7. Maybe one question while I'm getting the next
Anybody?
speaker's presentation.
Right. I'm happy. I can talk about this stuff all day. I love it.
You stumped them.
Yeah. All right. Well, I'll be around the rest of the day.
Thanks, Jim. All right, our final speaker, is this the wrong slide? For the session is Sarah Jameson. She's a service hydrologist for the National Weather Service out of Cleveland, and she's going to tell us about NOAA's focus on HABs. It's right here. Oh, okay, thank you.
Okay. All right, thank you. Is anyone here unfamiliar with NOAA? Okay, great. Want to put that up front there. I do work for NOAA, but I work for the National Weather Service. And I want to thank Jeanette for inviting me here in a week where you have a tornado, a flash flood, and snow.
You're welcome.
Welcome to Erie. Sidebar, I was working on Sunday. I was alerted to the flash flood warning for the area. And I was here on Monday assessing your tornado, classifying it as a tornado. So I've been up here quite a bit lately. But when it comes to specifically harmful algae blooms, NOAA is really starting to take off in this program. And again, I'm not a subject matter expert. People in this room probably know a lot more than I do. I'm a meteorologist, not a chemist or ecologist, so please be forgiving. I wanted to come here today because I am becoming more involved in the algae problem on Lake Erie. It's becoming the green monster in my life. Again, this is not something we were ever really taught, but because weather has an active role in the development of the algae bloom, I'm learning. And every time I go to these conferences, I'm learning even more. But be patient with me. Okay, so when it comes to NOAA's role in the HAB problem, as you can see from this map, this is not exclusive to the Great Lakes. This is a problem that they're trying to address nationally. And we do have some congressional authorizations to investigate into the algae problem. Our mandate is specifically for detection, monitoring, assessment, forecasting, research, education, and outreach for the coast and the Great Lakes. Who's not familiar with the 2014 water crisis in Toledo? Talk about shedding a light on a problem. And it's unfortunate that had to come to this, but this really did get a lot of federal interest in the algae problems. I'm also just recently joined the Western Lake Erie Drainage Basin Executive Board, again, where I'm learning a lot more about algae, where I get to hear about a lot of the federal resources trying to address this problem. And I get to learn a lot more about the problem and some of the things they're trying to do to mitigate it. But anyways, I digress. But anyways, this, again, really jump-started NOAA's role in the HAB program. Okay, detection and monitoring. I don't think I really need to talk about this given everything that I've heard in the room, but for those of you who may not be familiar, GLEROL, one of the big NOAA entities here in the Great Lakes, they have a lot of water quality stations. Ooh, I did the same thing you did. How did you do that?
Hit it again.
Hit it again. Okay, so where's the mouse?
The middle one.
The middle one. Okay, they're all different. All right, so here's just an example of one of the water sampling stations and the daily observations over time. As you can see, we have a fair number of stations. Now, these are just the ones that NOAA primarily maintains. But going to GLOSS, you can see that there's a lot of other entities that collaborate with NOAA. Limnotech, I work with you guys here, is another great resource of information on water quality monitoring and sampling. So it's certainly not just NOAA's role. There's a lot of other agencies that we work with. Okay. The beautiful Maumee River in Toledo. You can't talk about algae without this one stealing all the limelight. Not only do you have the largest drainage basin going into the shallowest part of the shallowest Great Lake, and it also happens to be about 90% agriculture. It's not a problem that's going to be solved overnight, that's for sure. But yeah, this was a picture of it this September, so it kind of speaks for itself. NOAA Event Response Program, NCOS, one of NOAA's many acronyms, National Centers for Coastal Ocean Sciences. They have basically a response team, and I kind of stumbled across this, but it looks like back in 2013, they responded to the algae bloom here in Presque Bay. So this is kind of like a short fuse, not a continual monitoring program, but basically on an as-needed basis, they can be deployed. I believe the funding is for less than $5,000 with these programs, but it is something that is available if there's a crisis situation. I don't know if anyone's familiar with this. I had not learned about this until last week. This phytoplankton monitoring network. In the Weather Service, we have spotters, weather spotters, and co-op observers who measure, rain and snow. Well, I guess we have a spotter program for people who want to measure algae and toxins. Anyone familiar with this program? I wasn't either. But when I showed this presentation to my counterparts at the National Ocean Service, they were more than happy to fill me in on this program. So they wanted me to promote it, but it sounds like we have a lot of observations already going on in this area. I'm not sure, how much of the National Ocean Service is involved with any of the water monitoring that you guys do in Presque Bay. Before coming here, I anticipated a question. And I'll get to it here in a moment, the forecast that the Weather Service tries to do for Lake Erie. And I'm like, well, why is Presque Isle not included in that bulletin? And yes, there is a scale difference. Obviously, Presque Isle is a fairly small watershed. But part of the reason I heard too had to do with the sampling sites, the need for sampling data. And after everything I've heard here today, I don't see that as a data void. But I don't know, this may just be a misinterpretation on my part, but it seems like maybe I need to communicate with you guys and maybe with my counterparts in Washington, D.C. about some of the information I just heard here today. Okay, research. I have no control over grants. I'm in no way involved with that. Okay, understand the drivers of the bloom ecology, which will aid in enhancing and predictive models that forecast bloom size, location, and toxicity. I think it's pretty well understood what drives the haves. I don't think that's really where the research is going into. It seems like the real problem is trying to predict, how the have is going to develop, and most importantly, the toxicity. We talk about the scale of the have, but really trying to predict anything in the amount of toxicity seems to be the biggest challenge, and that's where they're focusing a lot of the research grants and money into the forecasting of the toxicity. A few of the things that they use, in fact, this top right image, this robotic lab, that's something that just went in this summer. Again, this is geared more towards the Western Basin, but it's basically they call it a lab in a can, where it does all the work that you guys have been doing. I guess it's automatic. I can't attest to the quality of the data. This is just deployed, but it's great to see the real boom in technology and research tools. Of course, NASA's got a lot of things that they're working on, a lot of robotics and aerial spectral tools, and NOAA's heavily involved with that. Okay, this is where I get involved in the algae program. The Weather Service information from a few days to seasonal forecast and observational data gets used into a lot of what NOAA puts together in their forecast. Has anyone seen this before? Okay. Just wanted to see. So the early season forecast, it's a very technical product. A lot of your boat tourists, beach goers have a hard time interpreting these products. We're all scientists in this room, so it's, pretty easy for us. But what people want to know is how bad is it going to be? So the early season projection comes out usually sometime in May or June, and then they do a follow-up in July, because it's usually that loading that occurs during those early, late spring, early summer weeks that drive a lot of your sourcing. Okay, so here's just a look at one of the events. This was from this year. So we had a pretty heavy rain event right in the Maumee Basin, the worst basin to get the heavy rain, of course, the biggest driver that flows right into western Lake Erie. And after this rain event, looking at the bioavailable phosphorus, it shot up. And so that had a dramatic effect from the early season to what they were initially anticipating for the algae bloom. And that's how they kind of try to narrow this down a little bit. It's like a weather forecast. Well, it could be 40, it could be 60 degrees out, and then as we get closer, we try to narrow that down. So once we actually get into the bloom season, that's when we get the HAB bulletin that comes out twice a week. That's issued by the National Ocean Service. And again, you can see from these plots that it does not extend much beyond Cleveland. So at this point, it's not including Presque Bay. Discussions for another day. There's also Glarol, which has a HAB tracker. Has anyone seen this HAB tracker before? A few heads nodding. What I love about the HAB tracker, it's experimental, whereas the bulletin is operational, is that this you can actually see vertical profiles. So of course, if you're worried about water intake, what's happening at the surface is not as relevant as what's mixing down. So a lot of research going in there. You said it best. Data, 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 data. We can never have enough data sources. And here are what we use with the Weather Service. A lot of the forecast observational buoys that we have. Thank you for the one you guys put in over here. It's great. This information is used to drive a lot of what they put into the bulletin, because they need to know what kind of mixing to expect, what kind of currents, and we have the forecast models, of course, but we couldn't do it without the real-time data. All right, I'm going to go ahead and click this video because it's 4 o'clock and you guys probably want a little video. Let's see. Is it this one? Okay, here we go. Like I said, the Weather Service has kind of a limited role in all this HAB stuff, but we're kind of branching into newer areas, and this is one of the tools that we've come up with. This is just a little minute video. It's not on the audio. All right, while we get technical support. So when it comes to rainfall information, we all know how much of an influence the rainfall data is on not just the trying to forecast, but on the observational data. Again, I apologize for the loss of your equipment on Sunday. But when it comes to
responsibility,
sure, go ahead. I always say it's a money back guarantee with our forecast, so I'll take the blame on that one. Okay, it's working now. But when it comes to the rainfall forecast, obviously, if we get one major event, that's going to change any kind of a predictive model. And what's happened in Ohio is that they've come up with Senate one where there's regulations now on any kind of a forecast of a ½ an inch or I want to say 50% chance of rain. So here, let me go ahead and just play this now. This might describe it for you.
However, these nutrients intended for crops can find their way into our streams, and to reduce nutrient runoff, a multi-agency effort brought together the National Weather Service, universities, and other state and federal agencies. Together, they developed a tool tailored to the needs of farmers and nutrient applicators.
It takes into consideration soil moisture modeling, and so how wet the soil is going to be before that forecasted rainfall. It looks several days out, so farmers are able to see if today is or isn't a good day, if the next 2, 3, up to 5 days will or won't be better for them. And it also, during the winter time, takes into consideration models related to snow melt.
Protect our lakes, it'll protect our streams and rivers, and ultimately it'll do a better job of protecting large bodies of water, the Great Lakes, Mississippi River watershed, or even the Gulf of Mexico.
Okay, there's a much longer video if anyone wanted to learn a little bit more about that. The law in Ohio. Our role and what we thought was just a supportive role is now there's legal implications to our forecast and then how it's utilized. So this is an evolving, oh goodness. Okay, here we go. The role that we have is really expanding into the eco realm. And what I wanted to actually turn around and ask you guys, and we can do sidebars after the fact too, is clearly a lot of the research that you do involves some weather information. And, I'd be curious to hear how we could assist with the research community on providing you the best quality rainfall information, temperature information, lake temperature information, lake ice information. Now, granted, you might already have access to this information, but we would also like to see what we could do to try to provide that all in a one-stop shop. Because if you're familiar with NOAA, you know you have to do a lot of searching to find the information you're looking for. So we have a website on the National Weather Service office in Cleveland for the HAB. That's focused again on the Western Basin, but we can work together to incorporate some information that would help you with what you're doing as well. So we can sidebar that after the fact. Okay, future work. Go ahead. Essentially, moving forward, NOAA is trying to expand our role. We're trying to improve the models and the tools that we have to not only monitor, but actually predict. And I've seen tremendous improvement in it just in the last few years. So what they're doing is they're starting to expand the number of areas that they're trying to do forecasts for. So I think the work being done here in Presque, it seems like this might be the future just for this area. NOAA is always willing to work with state and local agencies. I think that's where most of you guys come in, to make new HAB forecasts. Okay, so here are some of the links to the information. I'm sure these will be provided after the fact for anyone who's interested in any of the stuff that we talked about, the HAB bulletin, some of the Galero HAB tracker, basically what I call the spotter program, the phytoplankton monitoring network, and the rapid response programs. And because I'm not a subject matter expert, I'd be happy to take some questions, but if I can't give you the proper answer, this is the team at NOAA responsible for all things HAB, and they're great. So I can go ahead and try to take any questions you might have. How much snow we're going to get? I'm open to anything. We got time. Am I the last speaker? So we got time. Want to ask me about the tornado track? Yeah.
What's better, the Farmer's Almanac or NOAA?
Microphone start. What? Well, actually, I will throw this out there. The predictions for the winter, and then subsequently into the spring, wetter. The trend is for it to be a little bit warmer, but I don't know if I believe that. But we'll go with that. But wetter. I don't like the wetter. Especially when we're talking about HABs. So, well, since we have a minute, I'm actually going to show you the webpage too. So if anyone else has any other questions. Okay, if you're looking for weather information, I'm sure that you all go to weather.gov/Cleveland. You can type Cleveland or just CLE. We actually have information right now at the top on the tornado event that just hit this. Was anyone near the track of this tornado? Anyone out that way? You weren't hit, were you?
No, it went right over my house, though.
Well, you're very lucky then. But actually what I wanted to point out here was the Lake Erie harmful algae bloom. Okay, so what we have on this page, and this is where I really am interested in input from people in this room, still loading, on how we can improve and add content onto this page. I have the USGS portal. It's loading. Here we go. This is the page where I really want to expand it, and I also want to have a section for climatology where if I can get recommendations from the research community on, for instance, what would be useful for you. Is rainfall information over a certain season? Is it weekly? Is it daily? Is it for basin? Is it for spots? What would be useful? You know, help us shape and design this webpage. That's what I'm looking for. So, some of the key areas of interest we have monitoring in Lake Erie, public water systems. This is where the USGS Nowcaster is located. Some of the information that you guys are producing. Oh, no, please go ahead.
You know, so one of the things that we always kind of have a hard time with, so if you were sitting there for my talk, I think, so I mentioned that mixing is kind of an interesting issue, and it's one of those kind of
hard to define parameters if you're not right there on the spot. So you can look at wave height, you can look at wave frequency, but some measure or of vertical mixing would be great. Because one of the hypotheses that I think a lot of people are working on is that when the bottom is resuspended back up into the water column, then you have a release of nutrients and potentially a reinoculation of cells. But there's not really a good measure except for wave height that would serve as kind of a proxy for that.
And is that uniform across like the lake?
No.
Presque Bay versus Sandusky versus the Western Basin.
In the direction and intensity of the wind, where it looked like within a matter of a week, we had a huge, maybe influx of lake water into the bay, and it caused a dramatic change in the plankton community. And I don't know if there's any way to kind of differently represent the wind data in terms of patterns and peculiar transitions in wind direction.
Well, I'm sure
that affects us in a big way.
Well, I'm sure there's a way that a climatology per se could be developed. Do they take water level readings here in the bay?
We have a level station installed at the channel between Presque Bay and Lake Erie.
Yeah, those are the kind of tools that we can put two and two together. We can start to develop a climatology.
What I don't think, I'm not aware of anything that predicts, that uses wind velocity and direction and speed to predict seiche events.
Well,
we can watch the history of the lake level changing at various locations up and down Lake Erie, but I don't know of anything that predicts that.
With accuracy? There are some models, and from the Weather Service perspective, we would do like, low water, and then flood warnings for, higher waters in the lake. So it's fairly rudimentary, it's not very sophisticated, but we do have some wave models that we use. Now, they're not detailed enough for like Presque Bay, but like Erie shoreline and things like that, we can, and I'm sure there could be a correlation from that. But there are some models. Again, the quality on Lake Erie, it needs forecaster interaction. And the Weather Service is actually developing what they call a total water. So for instance, like if you're on the coastline, if you've got a storm surge, in addition to your normal tidal action and high and low tides, they're trying to actually predict how high is the water actually going to be above ground level. Something that's always been a need, but we've never actually been able to produce. So it's the same models, even though we don't have tides per se. I haven't heard that coming to the Great Lakes, but I believe it is anticipated soon.
Folks who have boats and or operate marinas are very aware of the water level fluctuations hour to hour, as well as from season to season. But and we've had, of course, almost historically high lake levels this past spring. That's subsided a little bit now. But I don't know how I don't know how to quantify the impact on Presque Bay's water quality and ecosystem as a result of an influx of water from Lake Erie. Mike, that's what you were suggesting, I think.
Or the opposite, if the wind's from a particular direction, it could blow a lot of water through the channel out into the lake.
If Lake Erie goes up a foot, Presque Bay is going to respond over some time period to that, and there's going to be a tremendous amount of inflow through the channel. And then 6 hours later, that's going to go down.
I think there was an incident several years ago where they actually went up 5 feet in the channel.
I'm not at all surprised.
Yeah.
That's quite possible.
Within a short period, it went right back down.
Yeah.
Like a medio tsunami kind of thing?
Yeah.
Yeah. But no, these are some of the ideas that I like to hear, because there may be stuff that we have or are working on that could be incorporated.
Is it possible to monitor the water during the winter half of the year? The buoys that we operate here, the Regional Science Consortium has several buoys, but they come out of the water in the fall because they'll be destroyed otherwise. And so we don't have any data.
Now, what kind of data you're looking for, like the wind or water level or wave action?
The typical suite of water quality parameters that we routinely see: oxygen and temperature and conductivity and
I can't speak on those elements. I can say that the Weather Service will model all the weather elements. You know, the wind.
I'm not talking about modeling. I'm talking about just measuring. I don't know. I don't think that. I don't know how the weatherman knows the temperature of Lake Erie off the Erie coastline.
Oh.
In January.
Oh, that. Well, the Weather Service will call the water plants. That's usually where we get that information. If that's where the TV people are getting it, it's from us, then that's where we're getting it. Yeah.
And here at Erie, our water intakes are out in Lake Erie, not in Presque Bay. So that information from the inflow of water to the water treatment plant comes from a few miles out into the lake,
yes,
as opposed to our little harbor.
Yes, which the preference was, so that you're getting more of the lake water and not the shoreline. Yeah, I mean, we can house all kinds of information on our website. That would be helpful, even if it's stuff that you guys are collecting. If you're collecting water temperature in Presque Bay, we can archive that. You know, there's I think opportunity to collaborate. So just keep this in mind if there's a research study that you're doing and it just seems like there's some way the Weather Service may be able to help provide information or work together on. And I don't think I had, is my email address shared? Otherwise, I'm afraid I didn't put on the, I'll put it on the PowerPoint, and then when I get that shared, it'll be on there. So, okay. All right, thank you.
Well, that concludes the half session. Thank you to all the speakers. I believe that, what's that?
Anybody wants to stay and discuss this further, you know, future plans, maybe what everybody's doing next year, other issues, other things we can tackle, you're welcome to stay in the room and we can continue the discussion.
And the dinner this evening starts at 6 o'clock, so I think there's still tickets available if you don't already have one. Otherwise,